with that, we can focus on the science for the next 2 days. And so what I'd like to do in the next 20 minutes or so is to give you an update on all things PacBio with regard to the Sequel II system and so forth. So my talk has 3 parts. I want to tell you how it's been going with the Sequel II system, then tell you and focus the main part of my talk about HiFi reads and the 2.0 chemistry. And then I'd like to tell you about lowering the input requirements. Most likely I won't get to that this morning, and so if I run out of time, I'll talk about that part tomorrow morning in my intro remarks. That's the beauty of being the moderator of the, you can take your time and take your speaking slots. With the briefest of introductions, you know that at PacBio we aim to provide you simultaneously with high quality data, long reads, accurate consensus accuracy, least bias, single molecule long reads that are highly accurate, and methylation detection. And of course, initially that came at the cost of throughput. So initially the throughput was quite low, and so on the foundation of quality, we then worked our way up to increase the throughput, and the latest manifestation, of course, is the Sequel II system. This was launched formally in April of 2019, and the main differentiator to the Sequel, as you also know, is that it has a chip that has an 8 times higher feature density, 8 million features compared to the 1 million with Sequel. So it's been going very well. This is the, with regard to performance. I mentioned we launched in April, but there are data from the early access program going back to January, and you can see that in August, the average over all experiments of the global customer base, it was 140 gigabases per cell, which is about 9-fold compared to the Sequel, which is in blue. And so the reason why it's more than 8 is because the August data already includes some of the 2.0 chemistry early access that's specific to Sequel II, and then I'll tell you about in a minute. And then also with regard to reliability, we put a lot of effort with the engineers and the support teams, and you can see that the numbers are very good and continue to go down, both for Sequel and Sequel II, less than 2% of cell rate failure. So then with the traditional long inserts, you get a much higher throughput with Sequel II, of course, which depending on the DNA quality and the experiment tends to be between sort of between 80 and 180 gigabases. This one run is on the higher end. It's a 35 KB library, and you just get a lot more reads. So in this particular case, over 5 million reads. Half of the bases are in reads that are greater than 50 kilobases, and the longest reads are over 175,000 bases. So 8 times the yield, that translates to a reduced project time, lower cost, while maintaining the equivalent performance to Sequel with regard to read length accuracy and minimal bias. So just one quick example, and also highlighting our SmartGrant program on what you can do now and what this translates to. So this was our Plant and Animal SmartGrant winner from 2019, and this was awarded to University of Wisconsin researchers. You see Dr. Susanna Sample. And the hero of this story is Olympia, the German shepherd, who you can see here. And the SmartGrant was actually not conducted by us. It was sponsored by Histogenetics, and they did all the libraries, the sequencing, and the assembly, so we really had nothing to do with that. But you can see that 2 SMART cells were run about 90 to 100 gigabases each with very good insert lengths of about 21 kilobases. And so then the de novo genome assembly came out with a primary assembly of 2.4 gigabases, Contig N50 well over a megabase at 6.9 megabases, longest Contigs 34 megabases. And as you know, PacBio is unique in the ability to separate the haplotypes and express the genome in the 2 alleles. So a lot of the genome unzipped into the 2 alleles, about 2/3 of it. So not only you have one haploid representation, but you also immediately get the allelic differences with about 2/3 of the genome separating into the second allele, and also being highly contiguous with 3.1 megabases of the longest Contig in the second allele. And how does that compare, just as a reference point? Of course, here you see Tasha, the famous boxer breed that in 2011 the dog reference was based on. And you can see it's the same genome size, but about 26-fold greater contiguity and 20-fold less fragmented than that particular reference. So this speaks to the quality that you get right out of these PacBio assemblies. And so we're very pleased. By our estimates, there are now well over 1,000 eukaryotic genomes that have been sequenced with PacBio and de novo assembled. And we're very pleased to be a central part of a large number of large-scale initiatives like the Vertebrate Genome Project, the Earth Biogenome Project, the Darwin Tree of Life Project in the UK, and others. And this now extends to human genome projects. So quite recently, at the beginning of this month, there was the announcement that the SolveRD project, which is in Europe, a large initiative to solve rare diseases, has adopted PacBio SQL2 system to subject 500 human genomes and try to solve the underlying molecular causes of these diseases. And then just yesterday, the Human Genome Reference Project was awarded by the NHGRI. I'm sure many of you saw the announcements, and there were press releases by the University of Washington, UCSC, and the NHGRI to establish 350 reference quality human genomes in a pan-genome effort to finally finish the human genome for the first time and capture the ethnic differences in the human population to really get a comprehensive understanding. And the SQL2 system will have a very central part in both of those initiatives. And this is a good transition because these initiatives are not only interested in the traditional continuous long reads, but are particularly excited about the HiFi reads. So the HiFi reads, and then I'll come to the 2.0 chemistry, are, as you know, another unique differentiator of PacBio sequencing in being able to get you both long and accurate reads. And of course, this is afforded by having the SMRT template design where you have the adapters that makes the molecule circular. The polymerase goes into rolling circle mode. And in this particular configuration right now, the polymerase is very stable. It has gone around the template that it aims to sequence and made sure there's no nicks or no abasic sites or anything else. And it just keeps going. And so because of that, the entire read length distribution gets shifted. You saw in the first picture, the peak was to the left. Now the peak is to the right, which means most of the bases are in very, very long reads. In this particular case, half of the bases are in reads that are greater than 167 kilobases, with the longest reads going well over 300,000 bases. And so then you take advantage of the polymerase going around the SMRT multiple times, generating these subreads that have the random error and building an intramolecular consensus that gives you highly accurate long reads. How often do you have to go around to get to a certain accuracy? Not that many. 1.5 times around the SMRT, so 3 subreads gives you Q20 or 99% read accuracy, and 8 subreads, 4 times around the circle, gives you Q30 or 99.9% accuracy. And so that's what you end up with then is between 1 and 4 million, depending on the insert size, highly accurate long reads. In this particular example, the insert size was 12,000 bases on average, and the average read accuracy is now close to 30, Q30, 99.8%, but you can see that the mode of that distribution is really at close to Q40, and that's 1 error in 10,000 bases. So for a 12,000 base insert, that's like 2 or 3 errors in the entire read that is 12,000 bases long. So the utility of HiFi reads was first demonstrated on the SQL system, and this is recently published, so I'll go through that very quickly. In Nature Biotechnology, we collaborated with a large number of institutions to show that, in fact, you can trade off the read length from noisy long reads to shorter inserts, but having highly accurate reads. So in this figure that was made by Sergei Koren, he shows that you can get the same contiguity of a de novo assembly whether you use 70-fold coverage of continuous long reads at, let's say, 30 or 40 kb average, and the same you can get with 28-fold coverage of a 13.5 kilobase HiFi read or CCS read that are 99% or greater. And the benefit, though, is that you get a genome, a human genome, that's much more accurate than either the continuous long reads, which are in orange. So you can see in green are the CCS reads, and half of the genome is essentially perfect, so that's Q50, and that's measured over 100 kb chunks, so there's no error here. And then essentially all of the genome is better than Q40, and that's substantially better than the CLR approach in orange, where you get sort of average of Q40, and much better than what you would get with competing technologies, either by themselves or polished with aluminum. And so then, this is described in the paper, this high accuracy then allows you to call all the variants in the genome, not only structure variation, but also indels and single nucleotide variation accurately. It allows you to access more and map more reads to the reference genome. It allows you to fully resolve and phase into the 2 alleles, genes that are of medical interest. And on the bottom, it shows that you can now access a large number of genes that are inaccessible with the short read technologies and are medically relevant. And so this was done on a whole genome-wide basis. And of course, it has been utilized quite extensively already in the targeted area where you look at amplicons. And so this is just one example of an 11 kilobase pharmacogenomics gene that Stuart Scott at Mount Sinai studies. And you can see each line, whether blue forward or pink reverse, represents a single HiFi read. And you can see how clean the reads are, how easy it is to cluster them into the 2 alleles, how easy it is to recognize the homozygous and heterozygous SNPs and fully phase those variants, and not only substitutions, but also indels. Here, zoomed out is a 25 base insertion. And again, you can see how clean that data is. And just one other example, there was a study recently, for the first time asking the question, well, is it really relevant to sequence the entire HLA gene in order to improve the survival of transplant recipients? And the answer was quite astounding, that there is an enormous difference in whether you match them, actually match them, or they're not perfectly matched. So in this retrospective study, the researchers from Anthony Nolan in the UK looked at patients that they thought had been matched correctly, 12 out of 12. But then doing it again with PacBio, they found that in this bottom graph with the less survival, they were actually, in fact, not perfectly matched. And you can see that there is a dramatic difference in the survival to the patients that were, in fact, matched. So they highlight then in the abstract in the last sentence that this should be a technology that should be used going forward. And then it's also being used in a targeted fashion to identify disease-associated structure variants. So just one example, a study by Janet Song and colleagues from Stanford looking at bipolar disorder and schizophrenia and identifying in an intron a highly variable, highly unstable region. So in this colored plot that you can see down here, each row represents a patient, a human individual, and each of those colored vertical bars represents a block of this particular motif type, and the colors represent 2 variations within that motif. So just by the picture, you can see that there is enormous variability from patient to patient, and this is likely associated with the causing of bipolar disorder and schizophrenia, which has been studied for a long time and can now be recognized and characterized and further studied with PacBio sequencing. So there was a press release, hidden DNA sequences tied to schizophrenia and bipolar risks out of Stanford. And with regard to neurological disease and targeted sequencing, I want to encourage you and remind you and encourage you to submit. We have another SMART grant program, the 2019 Neuroscience SMART program. The proposal deadline is October 4th, but if you have projects that you would like to do, and a lot of these can be done on SQL, so this is still done on SQL, I encourage you to go to pacb.com SMART grant. We released a new protocol in the beginning of this month for amplification-free enrichment. There are some, especially in the neurological disease area, some regions of the genome you can't even amplify with PCR, and so we've developed a protocol, and I know many of you have heard about this. It's been a long time coming. We developed the protocol, and then we actually tested it and found that it wasn't as robust and as efficient as we wanted it to be, so we kind of went back to the drawing board and really reworked and started from scratch, as it were, but now it works really, really well. And so I encourage you to check it out. There's a webinar, there's protocols, and so you get really nice results. And as I mentioned, we're going to have a table and other breakout presentations if you want to learn more about that protocol. But now coming back to the whole human genome and looking at that with HiFi reads, others have looked at that. Evan Eichler and his team have looked at some SQL and SQL2 data for HiFi, specifically looking for segmental duplications, which are some of the most difficult regions in the human genome to resolve. And they find that with HiFi reads, you can get the highest fraction of resolved segmental duplications for any of the published assemblies analyzed so far, with an average 15% increase over even some of the competing technologies and assemblies that have been published. And the last sentence of the abstract is our results suggest that HiFi may be the most cost-effective standalone technology for de novo assembly of human genomes. Just one other example of how HiFi areas can even bridge really complex regions and complex regions. This is a collaboration with Dario Cantu from UC Davis looking at grapevines. And so he was looking at and sequenced the Cabernet Sauvignon and aligned the scaffolds to the Pinot Noir reference. And so in chromosome 7, there were 2 hyperscaffolds in blue and in red that still had a gap. And there was this contig in green from the HiFi assembly that in the self-dot plot revealed a highly repetitive 600 kilobases of alpha satellites that is the likely candidate for the centromere, showing that you don't need necessarily reads that span 600 kilobases or more because there's enough microheterogeneity in those centromeres, and the HiFi reads are accurate enough to resolve that. So essentially, if you have long and accurate reads, your repetitive content of your genome goes down because a repeat region that was 99.5% identical previously would be collapsed. Now with HiFi reads, you can resolve them, and it doesn't look like a repeat anymore to the assembler. So then, just briefly before I get into the new chemistry, just a couple of other mentions, and we're going to go into this in more detail in some of the talks. So Meredith Ashby, my colleague, later today will talk specifically about metagenomics. But just briefly, the HiFi reads are very useful to look at, for example, 16S sequencing. This paper, for the first time, to my knowledge, acknowledges the fact that bacteria have multiple copies of the 16S gene, and they're not identical. So this Staph aureus, for example, previously in the reference database, there was 1 sequence, but that doesn't reflect the biological reality of this bacterium having 3 copies of that sequence of the gene, and then it has 3 additional copies of the 16S gene, each of them having a different sequence. And this paper highlights that PacBio can resolve that. I encourage you to watch Luke Talon, who's also here, and gave a webinar. And I know this is a busy slide, so I'm not going to go through this, but just to show that with Sequel II, what that means is you can do a lot more samples. So on a single SMRT cell, he looked at 180 infant gut samples in 16S sequencing. This webinar was recorded, so if you're interested in that, go to PacBio videos and you can watch it there. And then with regard to shotgun metagenomics, the ability to get 5, 8, 10, 20 kilobase fragments from a complex microbial community and sequencing that with over 99.9% accuracy is very powerful, and we anticipate a lot of opportunities and a lot of growth in that area because you can get complete ORFs per sequence without even doing any assembly. And so that makes PacBio unique and also very powerful, in my opinion, to do high-resolution shotgun metagenomics compared to other techniques where you only get classifications on the family or genus level. With PacBio, you get to species or strains. Just a couple examples. We collaborated with Second Genome, looking at 7, 8 kb pieces of DNA from these complex community, sequencing that to Q30. And as I mentioned, there's now no assembly required for gene discovery, and they found advantages over short-read technologies with 2 times more unique error-free genes, 6.5 times more proteins of interest. So that really highlights the complementary nature. You see that now you get structural information, and of course the Illumina technology is still very powerful for counting and to get lots and lots of reads to look at abundances and get even the rarest members. And again, Luke highlighted another application for this for metagenomes, which is the ability to get complete genomes out of these complex communities. Again, I'm not going to go into that and refer you to the webinar, but he got a genome from one of these vaginal microbiome representatives in 5 contexts from less than 200 CCS reads. With regard to microbial with regard to non-microbial biodiversity, so eukaryotic biodiversity, the SQL system has been used, and there was a paper last year by the Biodiversity Institute in Guelph, Canada, to show that you can pool and sequence up to 10,000 amplicons in a single SQL SMRT cell, and we're pleased that this has been utilized by the Barcode of Life Initiative and both SQL and SQL II now. This was from a week ago where they got their SQL II system and tweeted in excitement, used for the BioScan initiative, a 6-year initiative to identify and characterize millions of species on the planet. And other work has been done before for museum specimens. So this is for herbarium specimens, PacBio metabarcoding, as well as towards PacBio based pan-eukaryotic metabarcoding using full-length ITS sequences, so expanding the region of interest to get even higher resolution. With regard to ISO-SEQ, we're also going to have breakouts and more talks about that. So very briefly, there was a very nice, very comprehensive review article by James Hatfield and colleagues just a couple of weeks ago, where over 300 references and more acronyms than you can possibly ever want to know. But it highlights very nicely the development of ISO-SEQ for the discovery of large numbers of previously annotated transcripts and detecting full-length homologous sequences across species. What's new with ISO-SEQ? Since late June, we have the ISO-SEQ Express protocol for faster library prep. This was a little bit cumbersome before, and it's now much faster, much more convenient. I've talked to many, some of you and many other customers. They really like it. And in combination with Sequel II, it improves the sequencing of longer transcripts. So you can see that compared to the template prep kit 1, the distribution is shifted to the right, so you get more long transcripts. And it gives you between 2.5 to 3.5 million full-length transcript HiFi reads. We've tested this in the bottom table. You can see here on a number of different tissues from human and mouse. And the conversion efficiency is very high, over 85% to get to 2.5 to 3.5 million reads. And in many cases, that's enough to fully characterize a transcriptome structurally to completion. So here's on the right a rarefaction curve, and you can see that it plateaus off. So in many cases, you can now completely characterize the structure of your transcriptome with just one SMRT Cell ADAM run. And there are other things that are a little bit more for the geeks. So in terms of the bioinformatics, 20% more genes are recovered per SMRT Cell. HiFi reads are enabled as the direct input, which makes it much faster. And the plot that you see here on the right is from a tool called Squanti2, which gives you the different classifications, novel isoforms, novel genes, and known splice junctions, and so forth. And then finally, IsoPhase is a software tool that my colleague Liz Sang developed, because not only is the genome diploid, the transcriptomes are diploid as well, of course, because you have two copies, and RNA is made from, most of the time, from both of those genes. And we can differentiate them because the sequence can be different. So IsoPhase takes advantage of the SNPs between the two genes, the allelic differences, clusters the expression, the cDNA, and you can see in this particular example that sometimes it makes a difference whether the gene is expressed from the mother or the father. So in the mother, this particular exon here is not spliced, and here you have a splicing event that's specific to the father copy of that gene, and you have an extra intron here. Single-cell ISO-SEQ is a very hot topic. I'm sure many of you do this. There's a number of papers that have come out that I don't have time to go into, but just to say that one example, this preprint here on the bioarchives, looking at influenza virus and asking what happens when you infect cells with an influenza virus. And the researchers found that out of the 150 cells infected, only a third of them were actually representing the wild-type virions. And there's a large number of different insertions, deletions, substitutions, synonymous, non-synonymous. So the virus is really bombarding the cells or the organism with a meta-viriome. And you need highly long and accurate reads and to do this in a single cell application in order to identify these. So there's a lot of utility in the HiFi reads that we've seen and that you have already applied. And because of that, we have put a lot of effort and increased the performance of that with the latest 2.0 release that's targeted to schedule next month. So I want to talk about that a little bit in the last couple of minutes. So what we found is that we still had in the chemistry 1.0 is an appreciable amount of these reads that terminate early and really don't go into rolling circles. So with the 2.0 chemistry, there is a substantial improvement in that, and so that you have more active polymerase reads, a greater efficiency of getting the polymerase into this rolling circle synthesis. That translates to an average overall 25 to 50% read length increase, and also allows to increase the insert size from 11 to 13 to 15 to 20 kb, depending on the application. As I mentioned, this is going to come out in October. We're currently in early access. So what you get then is, for example, for a 15 kb human library, 450 gigabases of raw yield, and again, 5 million reads. And then that converts then into a total of 30 gigabases of HiFi reads, 27 gigabases of reads that are greater than Q20 or 99%, And you can see again from the accuracy plot, the mean read accuracy is now Q34, 99.96%. I mentioned you can increase the library size. So here's two examples of a 20 KB library and a 24 KB library. So you can see still the yields are very high, 26 gigabases, 25 gigabases of Q20 and greater reads. And as the inserts get longer, you can see that there's a slight shift of the mean read accuracy, 99.92 to 99.8, but still a lot of really highly accurate reads, and now they're getting longer. For ISO-SEQ, half a terabase in a 24-hour run in terms of raw bases, that then gets converted into 4.3 million HiFi reads, full-length transcripts, 86% of those. Most of those, because of the short size, have no errors at all. So they're perfect reads, and 210,000 mapped unique isoforms from a single SMRT Cell 8M. Other improvements, of course, we always train the new chemistry, and so we have an improved base color that gives you about 5% more bases. As some of you may know, in the past, it's taken a long time to convert the raw data into the HiFi data, so we've put a lot of efforts into that and decreased the time that it takes by about a factor of 5 to 7. So you can see here that while initially it took over a week to get this converted, now it's about 4.5 to maybe 6 or 7 hours. If you want to do it in the cloud, it's even faster, 35 minutes for $70. So from weeks to minutes, and over $2,000 to $70 in 9 months, we've made a lot of progress. And we are targeting that with the release after this one, the next one in the spring, the HiFi reads are going to come straight out of the box. So there's no more computation time or anything that's required, but the HiFi reads will come straight to you as the user, and the time and cost will be zero. We've updated the workflow engine. SmartPipe admittedly was not the best workflow engine, so we've implemented Cromwell, which was developed by the Broad Institute. It's widely used, it's open source, it has smart recovery after job interruptions and so forth, and runs in these hardware agnostic WDL and CWL languages that the industry is using and is standard, and makes it a lot easier. And then finally, with regard to the HiFi-based de novo assembly, it's now available with Bioconda, the FALCON UNZIP 3 using the HiFi reads, so going straight into the HiFi reads. I mentioned speed, so FALCON UNZIP has been modernized to use HiFi data and modern tools. And you can see that it's quite a significant reduction both in the data storage requirements, so this is the file size, by about a factor of 3 compared, this is on a human genome, by about a factor of 3 times less storage, and by about a factor 2 times less of the compute time, whether it's CPU hours or wall hours, that's scales. And so there's a twofold improvement. And then, as I mentioned, eventually this is going to go away, and that's going to give an even higher improvement when the HiFi reads come out straight out of the machine. And it's been optimized for accuracy. So you can see here the comparison that with the 2.0 chemistry, again on the human genome, the accuracy is now Q50, where it was with the CLR, the highest that we've seen is 45. So the HiFi reads are now, with regard to assembly, both more contiguous and more accurate than the continuous long read assemblies. And this is thanks to also the speed up and the improvements in accuracy are a new consensus caller, not Arrow anymore, but we're using Racon. And so here you can see what that translates to, both in terms of contiguity and accuracy. This is a human genome. The contigs longer than 20 megabases are shown in red. 6 of the chromosomes spanned by 2 to 3 contigs. And so this nicely corroborates what's been shown and observed by the community, such as Adam Filippi's group, that when you are able to separate the haplotypes, you can get very high accuracy, and there's no more polishing of short reads required. And then with regard to phasing, the haplotics are already phased, and so you can see this here, where you have parental information, dad is the y-axis, mother is the x-axis. They're already perfectly phased. The primary contigs, of course, by design, are still mixed haplotypes, and so there you can use tools like the trio binning if you have parental information, or if you don't have parental information, we've developed a tool in collaboration with Phase Genomics called FALCON Phase, which combines with Hi-C data and can assign and phase the primary contigs as well. And the nice thing is you can do that on the contig level to get to phased contigs, and then after scaffolding, you can run it again at the scaffold level to get to chromosome scale phase diploid assemblies. There's a preprint in the bioarchives for more detail about that. So then just lastly, for plant animal assembly examples, here's rice and Drosophila, both showing that comparing the CLR approach to the HiFi approach, this is a greater than 30 kb library compared to a 17K library, 60-fold versus 20-fold. The contiguity is about the same, or about twice for the Drosophila, and you get really good yields, 15 gigabases, 31 gigabases, way higher than you would need for one rice or one Drosophila. So we're working on multiplexing multiple samples, barcoded samples, into a single SQL2 cell. We're again collaborating with the Sanger Institute on that to have even more cost-effective solutions for these types of genome sizes. And just for completeness, other improvements, the HiFi library prep is now using the Express protocol, so that was the last application where that was moved over to the Express protocol. And then, as Meredith will talk in more detail, SmartLink 8 now has an end-to-end multiplex microbial analysis, including assembly, circularization, and plasmid identification. So SQL2 then allows to have you a lot of things that you can do with just one SMRT Cell. And so we now have documentation where the different applications are highlighted with the relative throughput that's needed and the prices that that's going to take. And we believe because of that, the SQL2 system is very attractive for a lot of different applications, for core labs that's of interest, and for a lot of different researchers. So with that, I'll close and highlight that there is a webinar that's going to happen on October 9th that I invite you to attend. And then the last, and you know this is my acknowledgement slide, and I showed a lot of the work that was done not by us, but by all of you and by the scientific community. So we feel very privileged and grateful to be involved in the work, and we are so grateful for all the time and efforts that you spent applying the technology, pushing it forward, and working together. And so I really look forward to the discussions and all the presentations over the next 2 days. So I did run out of time, and so I'll take more time tomorrow morning on telling you about the latest developments on lowering the input requirements. But with this, I'll close. And I think in the interest of time, we will just, yeah, we're not so bad. We'll just continue. I'll be around. And so with that, I will close and pass it on to the next speaker. Thank you very much.